Great. Well, hello, everyone. I know we've got folks joining from both coasts and in between tonight. I know many of you on the Zoom webinar today. For those who don't know me, my name is Dr. David Caro. I'm our company's president, president of HLI, as well as our medical group. And it's a great honor today to introduce Dr. Robert Green. I'll say a few words about Dr. Robert Green's bio in a moment. But just to kind of tee up today's conversation, I think many of you on the Zoom I know are longtime clients, and we're grateful for your continued support. But we, as you know, had our beginnings, our humble beginnings in the clinical genetics field, going back all the way to the year 2000, when our co-founder, Dr. Craig Venter, helped sequence the human genome, along with Francis Collins and the NIH effort. And from there, we really, as we thought about what we could do in the precision medicine space in our first clinic here in San Diego, now two clinics, one in the Bay Area, we really thought about, how could we together reduce precision medicine into clinical practice. The promise of the genome sequencing effort was that it would usher in a new era of personalized precision medicine, preventative, personalized, and genomics based. Now everyone, of course, is talking about AI machine learning based, but the whole concept is how can we use data, genomic data, along with phenotypic data to not only understand the current health status of an individual, right, but also predict many years out what is their pre-symptomatic risk for all the chronic age-related diseases. Along the way, Dr. Tom Caskey, who is really one of the pioneers in clinical genetics and started the clinical genetics program at Baylor, which is still alive and well today, joined us as our chief medical officer. Tom recently passed away, and he really had a huge imprint on all of us and certainly on our company. So with that, let me just say a few words by way of introduction to Dr. Green. Dr. Green and I've gotten to know Robert personally, and it's great to, again, have you advise and counsel our ecosystem today. He's a professor of genetics at Harvard Medical School and the director of the Genomes to People Research Program at Mass General Brigham and the Broad Institute. His pioneering research in genomics and precision medicine has set the stage for global advancements in preventative healthcare. As a leader in the field, Dr. Green's work is focused on how genomic information can be integrated into clinical practice. He's authored numerous papers on the implementation of genomic screening in healthy populations. So, Robert, I'll let you take it away.
Thank you so much, David. And to everybody, it's a pleasure and an honor to be here. I got to tour HLI way back when it first got started. I got to know Craig a little bit and was recently on a platform with him and have been on a number of platforms with him over the years. And I just think what you have been doing is so much the future. And maybe think of me as sort of the one of your academic wingmen as you're trying to convince the world through evidence that proactive and preventive health is the right way to go. So I'm going to share my screen and show you some slides. And we go. And now I'm going to go into presentation mode. So is that showing up okay?
Yep, looks great.
Okay, great. So I'm going to talk today a little bit about preventive genomic screening for longevity. And it's a little bit deceptive because I'm not going to get into the weeds about longevity science. I bet you're going to have lots of speakers on that. But I'm going to talk about it because in this moment in time, in this moment in history, one of the best ways to live a long time is not to die early. And that's really what so much of what you're working on at HLA, and it's so much of what I'm working on through the prism of genetics. I'd like to mention my sources of support, mostly NIH grants. Thank you for your tax dollars. Also some additional foundation grants. And I do compensated advising for a number of companies there and am a co-founder of two companies, one of which I'll tell you about at the end of my talk. So again, this is a very sophisticated crowd. But let's quickly remember where the science of medical genetics is at this moment in time for most people. And at this moment in time, the vast majority of geneticists around the world are busy their days by diagnosing children who come to them, primarily children, but sometimes adults who come to them with mysterious conditions, by doing a limited amount of preconception screening for couples who know they want to have a child, by doing some pre-symptomatic testing in families with a genetic diagnosis. You know that there's cancer in your family, or even you know that you're carrying a BRCA mutation in your family. Um, some child who gets a positive on conventional mass spec newborn screening in the state mandated public health programs and needs some genetic follow-up. Prenatal screening, principally for chromosomal abnormalities and structural variation through the NIPT, which is taken off in pregnant women. And then a huge, huge area of targeted therapies for cancer, where we're actually sequencing the tumor, not the human being. And so this slide is kind of the world of everyday genetics in most of the country, even in most medical centers today. But as you've had the advantage of at HLI, and as I've been working on in my clinical research project, and as the world is coming to appreciate, there's a lot more going on. There's the idea that we could look at the entire population and we could screen the population for risks. There's doing that even more intensively in newborns because newborns have can have a whole bunch of childhood diseases that are many of which are treatable. HLI is already getting pharmacogenomics and polygenic risk scores. So you know that those are adjuncts to the conventional monogenic approaches. There is a number of people working on prenatal sequencing. Imagine taking a sample of the mother's blood, being able to find that needle of DNA, of fetal DNA in the haystack, and sequencing that, and then really expanding carrier screening so that there are more standards, that it's done increasingly well, so that as few couples as possible have to go through the horrible pain of having a devastated child. And of course, where we're going is not only gene editing as a kind of science fiction tool, but it's already being used for treatment. And there's this whole idea of the big data integration, which has been around for over 10 years, the big data integration of the genome with transcriptome, proteome, metabolome, microbiome, all the exposures, all of the things that you can add to it. And I don't know everything about HLI, especially recently, but I do believe that HLI is an example of an attempt to do this with folks to layer in as much of this data as possible and have a data-driven approach to health. And so I applaud you for that. And I believe, as you do, I think that this is the way we're going. Now, when people with resources in concierge practices and in HLI can get their genomes sequenced, but why is it not widely in use? And I would say that this is the strange case of an entire specialty that's a little bit afraid and has learned to be afraid of its own technology. The notion that genetic information is toxic, that it's somehow going to harm you. The notion that there's going to be widespread privacy and discrimination issues, the notion that participants and especially even their providers will completely misunderstand what's going on, and the idea that the harms and costs are going to outweigh possible benefits. These are powerful narratives that have been passed down from trainer to trainee in the apprenticeship system that is academic medicine for decades now, and I believe that they are fundamentally flawed, not in the sense that they're wrong. They're all important questions, but in the sense that we have over-indexed on these questions and under-indexed on the life-saving potential. Now, I'm fortunate enough to work with a lot of people in my environment here in Boston and around the country who are interested in exploring these issues. And the focus of ours is essentially the risks, the benefit, and the cost of genomic information. We are an implementation science unit, and I do see patients, but what I do most of all is run large teams of research groups that are trying to answer these questions either in single site studies, multi-site studies, or broad studies. Now, we were among the first in adults coming out just about the same time that Craig published the first HLI paper in the journal on the results of the first folks that he did comprehensive sequencing and return of results on. We did something like this too. We called it the MedSeq project. And we published the notion that 20% of you who are listening to this are carrying a dominant mutation. Now, that doesn't mean you're going to get the disease. You might die before it manifests, or it might be only incompletely penetrant, meaning it never is going to manifest. But by virtue of carrying that, you are at risk for certain types of cancer, certain types of cardiomyopathy, certain types of sudden cardiac death, even weird things like skin diseases, eye diseases, ear diseases. And so if you look hard enough, if you look at 6,000 genes, you find this in people. I think we all recognize that almost everybody's carrying recessive mutations that can lead to affected children. Almost everybody's carrying an atypical response to at least one category of meds. And if you think about polygenic risk scores, if you think about 6 or 7 or 8 of them at least, almost everybody has got an elevated polygenic risk score. So taken in the aggregate, there's a lot of information in the genome. Now, I usually spend more time on this in adults. I'm not going to spend that with you guys because I think you're already believers in this. But I do want to emphasize that the yield for testing adults very much depends on how many genes you analyze. This is probably intuitively clear if you think about it, but many people haven't really thought about it because they think that when you get whole exome sequencing or whole genome sequencing, that is equivalent to looking at all the genes you've just sequenced, and it's not. So whether you get a panel or whether you get whole exome or genome, if you look at just say 10 genes, you find 1% to 2% of the population that's carrying a dominant mutation. In 2013, I led a group that created the first list for screening, and believe it or not, the only list for screening genes that's ever been created by an official body. This was the American College of Medical Genetics incidental or secondary findings list. And our first version of that had 56 genes, the second version had 59 genes, and the third version now has 81 genes. And the importance of this is twofold. One, it's the first time any learned body has endorsed any form of genomic screening. The CDC Tier 1 was not an endorsement of screening. It was an endorsement of cascade testing when it came out, although it's widely misrepresented as screening. Since then, of course, many groups have decided to create their own panels. Invitae created a panel, a very sensible panel with 167 genes. HLI uses a lab that has its own panel. So whenever you send the genome for screening, you got to ask how many genes and which genes are they examining, and have they validated for those genes, not just in terms of single nucleotide polymorphisms and indels, but have they actually validated to make sure that they can detect the structural variations and various forms of mutation in the genes they claim to be analyzing. And in our studies, MedSeq, the first to comprehensively sequence adults, BabySeq, the first to comprehensively sequence healthy babies, and MilSeq, the first study to sequence active duty military in the Air Force, we looked at between 4,000 and 6,000 genes in these, and we have consistently found monogenic disease risk in between 16 and 20% of people. Just think about that for a second. That's incredible that there is so much information right there. Let me just cough for a second. Okay. Now, where we're heading with this is the notion that we need not necessarily wait till we're older and to get ourselves sequenced, but really genomic information should be available throughout the lifespan. And in this sort of virtuous cycle where we can do preconception testing, we can do prenatal testing, we can do expanded sequencing in newborns, we can look out for any risks that a child might have and even start that child on interventions that will lower their risk for the future. In young adulthood, we can know if we are at risk for high risk cancers, or we can gradually work in polygenic risk scores and pharmacogenomics as our age grows, and we would be faced with those conditions and those drugs. And we would be constantly updating this. And at the end, I'll tell you about a startup company that we've recently formed to try to manifest this vision. But it is really the vision that we've all been working on. And in my academic career, I've approached this from three different vectors, if you will. And you might find it interesting to think about how do you approach the idea of DNA first? You're an academic, you got to write grants for a living, you got to write papers. I don't have the luxury of just opening a shop and saying to people, come, because I think this is a good idea, come get yourself sequenced. So for the first part of it, there was a huge 10-year debate about incidental findings. If I sequence you for a heart problem and I've got your whole genome, what else should I look at? What else should I just stumble across a BRCA mutation, an incidental BRCA mutation, or should I go after and hunt for it? What should be the standard? And there was so much going on about this. And I was very lucky because I started in genetics late and I didn't have enough enemies for anybody to block it, that I got to lead this ACMG recommendation that I mentioned to you before. And this is where we came up with 56 genes that has eventually come 81 genes. And this was a real breakthrough. It was a crack in the door because I enlisted all these experts who had great credibility and said, you know what, maybe genomic screening could be useful. And then there was the return of findings. Look, we've been sequencing millions of people as part of research studies, but for almost none of them do we give back an iota of information. Is that fair? They've given us their DNA. Most of them actually tend to have an idea that if we might look for something, we might found something, we might give them back something that's useful for them. And in fact, we typically don't. So I wrote the first grants to return research findings in, say, the Framingham Heart Study or the All African American Jackson Heart Study, and or our Mass General Brigham Biobank, and sort of set the standard along with other investigators like Susan Wolf around the country in why you should do this, what you should do, and how you should do it. So here's one of our papers on that. And then what you guys, those of you who are patients and those of you who are working at HLI, what you guys are doing might be called elective genomic screening. You've elected to get it, or population screening. If in this case the population is the members of HLI. And how does that work? How can we get that going? We've actually even written some early work on that, where we said, let's see what happens when you do this and you work through primary care rather than even working through geneticists and genetic counselors. We kept genetic counselors out of the picture. So these are three sort of flagship articles, but we've written almost 100 or more articles on these themes, and together been trying to socialize and create the evidence base for DNA first. Now, I want to really drill down on this talk into the question of newborn sequencing, because as you'll see from our papers, and even today, I believe a paper just came out in JAMA Open Network on this topic, and it's a very hot topic that is finally exploding around the world. And I want to tell you the story of this topic. So you'll see Thomas Caskey, one of your founders, statement from 1992. that newborn screening would eventually be expanded. And other notables through the years have made these similar statements, including Craig. Within 10 years, every baby born will have its complete genome repertoire determined. Okay, so that hasn't quite happened yet, but as you'll hear, I think it's starting to happen now. The President's Council on Bioethics, Francis Collins, Alan Guttmacher, and I've gotten to be friends here in Boston with a remarkable fellow, Wally Gilbert, Nobel Prize winner for the very first human sequencing of DNA, the very first sequencing of DNA, even before Sanger sequencing. And he says that he and others were talking about this in the 1980s. So um, this was an old idea. And in 2013, we wrote a grant, which we nicknamed the Baby Seek Project. And we gotten it renewed from NIH. And for 10 years, we have been almost the only ones in the world who are crazy enough or bold enough to sequence apparently healthy newborn babies. This group has now expanded to over 100 investigators. And the first question we asked, of course, was, do parents want it? And it turns out that not just educated parents, not just wealthy parents, not just tech-savvy parents, but most parents, if you frame this in that, would you like your DNA baby's DNA sequence to discover treatable conditions? Almost 70% reliably say they do. Now, I told you about screening genes, and if you start a project like this, you got to decide with a blank slate of paper how many genes you actually want to include. So we went through a whole process of thinking about the validity of the gene disease association, thinking about the evidence, because there's a lot of papers out there that probably aren't correct. Thinking about the age of onset of the conditions, thinking about the penetrance, the probability that if a child carries a mutation, they're actually going to get the disease. And we came up with initially with 884 genes, and then very controversially, we added back 70 genes for cancer predisposition in adults, ending up with about 1,000 genes. And so in this initial list of 1,000, we actually have gone on over the years to expand this. And our current baby seek list is we're looking at 4,299 genes at the top of the funnel. And then we have a dynamic process that in real time analyzes disease validity, pathogenicity, age of onset, and penetrance, and spits out at the other end of this algorithm with a minimum of human intervention the variants that we think are pathogenic and likely pathogenic. And they are mostly neurological, if you just break them down by gene number. A lot are metabolic, a lot are endocrine, a lot are cardiovascular, and then a whole bunch of other things. Now, remember, when you look at a gene, there's a lot of changes in the gene. We call these variants. The old word for them were mutations, but we call them variants today. And we distinguish between pathogenic variants, variants we think do harm, and benign variants, variants that don't do any problem. And for BabySeq, we reconceptualized what it means to look at variants. So if you're in the diagnostic space, if you got a sick child in front of you and you're sequencing a child in order for cause, you don't really care about clinical actionability. You're trying to make a diagnosis. You're going to look at pathogenic, likely pathogenic, and variants of uncertain significance because you're almost learning from that child and that family as if they were research. And for that reason, by the same token, you're not necessarily only looking at well-established disease gene validity. You're looking down the scale here at questionable things. So you've got a larger search space, if you will, for indication-based or diagnostic testing. But when you're thinking about screening, you want to lean into specificity, not so much sensitivity. You can actually afford to miss a few because your goal is not to capture every single person who might possibly have a mutation. Your goal is not to miss things that are definitely harmful for that child and that child's future. Because if you lean into sensitivity, you're going to identify so many children that aren't going to ever get sick, that you're going to send the whole healthcare system into a deep spiral of over medicalization. And then we designed a report with just a little bit of education. We felt that even any pediatrician could read. And then we thought, well, this is so controversial. And remember, this is 10 years ago. How do we design this protocol to satisfy the world? And so we designed this initial protocol as a true randomized controlled trial to ask what are going to be the benefits and the possible harms of doing this. So we got volunteers who had just had a baby, and they signed up, and then we randomized them to get sequencing or not. And let me tell you, when you people who are volunteering to get their baby sequenced, and you tell half of them they can't do it because it's part of a research study, they are not happy. But they understood, and we went forward and we measured the medical, behavioral, and economic outcomes in this study. And we screened the first infant that's ever been done as part of a research study that I'm aware of on May 22nd, 2015. And since that time, we're coming up on approximately 1,000 families. And this is the initial group that screened that first baby in 2015. Now, when we got done with our two IRBs that were supervising us and our extra advisory board, we had a consent form that was so onerous that when we asked people if they wanted to be sequenced, if they wanted to participate in a research study that involved DNA sequencing, most of them declined without even listening to what we had to say. But when we sat down with them and they gave us time and they listened, about 10% of them said they wanted to hear about it. And again, about 2/3 of those did enroll. This paper has been really interpreted. It's like a Rorschach. Some people have interpreted this paper, especially at the beginning, like, you see, nobody really wants this. It's a bad idea. But what it really is that when people have just had a baby, like just had a baby, they don't necessarily want to go through a 22-page consent form. Not fun. And what did we find in that initial yield? Well, amazingly, even with about 1,000 genes, we found 11% were carrying monogenic disease risks. And these were not just things that they were at risk for the future, like dilated cardiomyopathy, hypertrophic cardiomyopathy, hemolytic uremic syndrome. But amazingly, these were things which were happening right now in that child. This third row, for example, this elastin mutation, had a perfectly healthy baby. And when the baby had an echocardiogram following the discovery of this mutation, sure enough, they had a abnormally narrowed aorta. And that was stunning to me. We weren't just talking about the future. We were talking about the present. And by the way, this is what your paper early on, that paper from HLI found as well, that when you found people with cancer predispositions and you looked harder, you found some people with cancers that were already there. One of the huge narratives in this whole thing is, in adults, you're going to terrify adults, or in children, you're going to terrify their parents. And that's where the strength of the randomized clinical trial really came in, because we could literally compare the same type of people who were volunteering and who gave themselves, who got sequenced or didn't get sequenced. And sure enough, there was no perceptible difference, no measurable difference on validated scales of depression, anxiety, self-blame, or parent-child relationship. And what about the downstream economic spending? This is a real touchy subject because it speaks to social justice, it speaks to the solvency of the healthcare system, it speaks to a healthcare system where there are gatekeepers who are, in most cases, who are insurance companies. In terms of groups like HLI, who have members who can afford to spend more for downstream medical, does that give them an advantage that's fundamentally unfair? Are we widening healthcare disparities? But But when we looked at this again with the randomized clinical trial, it turns out there's so much noise in the healthcare spending of the first year of life that the difference, we didn't count here the cost of the sequencing, but after sequencing, the differences weren't particularly meaningful at all. So that's kind of reassuring. And even when you compared the spending on the children who had a monogenic disease risk and the children who had no monogenic disease risk, there was some difference, but it was not wildly different. And it was mostly justified. It was follow-up for echocardiograms, for dermatology visits, for screening that had been indicated from the mutation that was found. Now, a big question we always get is, are the results actionable? And most of the studies that have now followed us in the world have only looked at actionable conditions, meaning conditions for which there is a FDA-approved treatment. And that is the prudent thing to do. And in fact, that's what we're doing in our company. But in this research environment, we wanted to lead the way and ask, what do you find if you look for everything? And how much of that is actionable in one way or another? And we were stunned. We use this semi-quantitative scale that's been published by others to sort of as a proxy measure of the concept of actionability, like how severe is the disease, how how penetrant is the disease if you have a mutation, how efficacious is the intervention, how good is it, and how tolerable is it? So adding a vitamin is very tolerable. Doing risk reducing surgery like mastectomy is a pretty difficult to tolerate intervention. So with this, you get a kind of full diamond shape that represents sort of good actionability. You get this odd triangle shape with a disease that is not actionable, like Huntington's disease. So now let me show you the shapes of all the things we found in BabySeq. Remember, full triangle is good. I'm sorry, full diamond's good. Small triangle is not so good. Well, here are the shapes we found for all of the conditions that we found. None of them are fully unactionable. And by the semi-quantitative criteria that we used, every single one of these conditions was actionable. Now, there wasn't necessarily a pill, but there was always something you could and probably should do in terms of surveillance and so forth. Moreover, we, as is so often the case, but is so often under emphasized, when we found each of these babies in the top row had a mutation, turned out that each, since these were almost all dominants, there was always one parent with the mutation as well. These are not de novo mutations, these were inherited mutations. And it turned out we were able to shed light on medical conditions that the parent had that they had no idea was due to the genetic condition. So only 17% of our infants were found to have a phenotype defined as a clinical feature or an X-ray feature of the disease, but 23% of the parents who never knew they were carrying that mutation were found to have a related phenotype. And if you count the family as a unit, that means 35% of these family units were found to have at least one person with a related phenotype at this moment in time. Okay, at this moment in time, not even the future. That's incredible. We are finding manifest disease in 35% of these families by checking the DNA of one baby in that family. And because we were brave enough to do a few adult onset conditions like BRCA1 and 2 and MSH2, which is part of Lynch syndrome for colorectal cancer, we found three mothers. By coincidence, could have been fathers, but three mothers who themselves were carriers of the same mutation, none of whom knew it. None of whom knew it. And then went on to realize their whole families had these mutations. And all three of these mothers in the 5 years since this discovery have now had risk-reducing surgery. Probably the greatest contribution of BabySeq to these families was that we saved these mothers' lives. And that was absolutely not an expected outcome when we started this research study. Let me show you a couple of the voices, and these are not cherry-picked, the voices of some of the parents. And I'm hoping you can hear that okay.
Yeah, Robert, I can't hear it. Um,
Oh, oh, you can't hear it?
no.
Okay. Let me try sharing one more time. I did share with sound. Let me try one more time to share with sound. See if I can make this work. Okay. Okay, so the for some reason this Zoom is not letting me select the option to share sound. So we'll skip this slide. But basically, the sorry. So basically, the video shows a couple of parents saying that they really appreciated it, including one of the mothers who had risk-reducing surgery, saying
Oh.
that she's willing to be around longer. She's now able to be around longer for her child. So BabySeq is expanding now into multiple sites. And if any of you happen to be associated with pediatric practices or academic centers that might have a pediatric champion, we are looking for additional sites around the country, particularly sites that, not exclusively, but particularly sites that can recruit diverse families. And we are talking to people at both Stanford and UCSF and USC. I don't think we're talking to anybody in San Diego, but we'd be delighted to if that's an option for some of you. And we're able now to show that we're recruiting not just folks of Northern European ancestry, but non-European ancestries, Spanish-speaking, lower educational, lower income level. So we're really demonstrating that this doesn't have to be something that's only attractive. Obviously, we're paying for it; they're not paying for it, but it's not just attractive or used by folks who are more privileged. And in BabySeq 2, with this much more diverse cohort and a larger number of genes, we are continuing to find 10 to 18% of babies with a monogenic disease risk. So this second cohort is finding it. And because ancestry is such an important bias in genetics, I know you're aware of this because you're doing polygenic risk scores. The question of, for example, polygenic risk scores in different ancestry groups has not been solved yet. Many people are working very hard on it. But we are looking very carefully at our folks from BabySeq 1, which was not very diverse, and the folks from BabySeq 2, which is very diverse, and looking to see if there's any major differences that we can see. And this is sort of proof of concept because the numbers are still small. We're not into the thousands at all. But so far, we're able to give back an approximately similar number of monogenic disease risks. Now, before I finish and open up for comments and questions and discussions, and I'm really interested in your thoughts because you're a very engaged group here. I want to tell you about a startup that we've just started 2 years ago called Nurture Genomics to try to scale this for the world. And Nurture is building on this decade of research, which has got a lot of media attention that I've worked on. It's a HIPAA compliant enterprise level PCI ready protected web app for both desktop and mobile. And it is actually up and running for friends and family, and for any of you who would like to try it, for children of any age in California, Texas, Illinois, New Jersey, and Florida. No advertising yet. We haven't officially launched, but the DNA is collected with a kit that swabs the child's cheek. It's sent to the Broad Institute for best quality uh, sequencing and an analysis of 413 eminently treatable genes to start off with. That's our V1 product. And then just as you would with any product like this, you follow. And anytime you can have questions with the genetic counselor. So we are a medical practice as well. We've already been talking. We haven't been accepted in all these, but we are in talks with all of these pediatric practices to begin to present this to their patients. And here's the website for nutrigenomics.com. If any of you would like to go on and use the friends and family discount, it'll be available probably only for a few more weeks at a price point of well under $1,000. And then we'll eventually go up to a much higher price point because all of this is very hard to drive down. But we just graduated from an AI incubator, and we'll be bringing some about $1 million worth of AI technology to bear on driving innovation and the price down. So if anybody's interested, I'd very much like to talk to you about that. We are rounding out our pre-seed investor round with a cap discounted safe for investors, and we'll try to finish that before the end of the year. But meanwhile, here are some of the web links for you if you're interested. Our research program is genomes2people.org. Nurture Genomics is there. We're on X at these handles. We're on Twitter, and here's my email at both Harvard and at Nurture. If you'd like to contact me, I'd love to hear from you. We accept brilliant students and trainees, mostly college and medical students, but occasionally a precocious high schooler, to work with us over the summer. They love it because we're doing real clinical research, and we have a very cohesive and fun group of people who we work with in the Boston area. And we even take some students who can work with us virtually without even traveling to Boston so would love to speak to any of you who would like to write me at that email address. So at this point, David,
Yep.
that's about 45 minutes. Let me stop. I'll stop sharing, and you can moderate the discussion.
Yep. No, thank you so much, Robert. That was incredible. I learned so much and learned so much since we last spoke in person. And I already see that we have a bunch of questions, and I'm sure there'll be many more. Folks, we do as we always do. We'll end kind of right at the 30-minute mark, so in about 15 minutes. But I'll try and get to as many questions as we can. Not surprisingly, the first one, of course, was okay. Well, how many genes does HLI assess as part of their whole genome? Which is a great question. And actually, the answer is it depends, right? Because so HLI actually is part of the R&D, research and development we've had over the years. Developed a tool called HLI Search. And there's a fair amount of intellectual property behind that. And underpinning HLI Search is about 50,000 genomes that we've sequenced over the years. And that really allows us to look, gene by gene at nearly every known gene. And the way that we use that is when there is a phenotype. So someone comes in with an extensive history of breast cancer, or a client comes in who herself has presented early with breast cancer. But yet the automated reports, whether it's ours or Invitae or some other exome report, showed nothing. We then can do sort of what we call a deep dive and really look beyond what is in the CAP-CLIA sort of automated report and see if there is anything that pops up. And actually, more often than not, when you have a dramatic phenotype, especially over multiple generations, not surprisingly, you do find it. Often it's a VUS, a variant of unknown significance, that has not yet made it to ClinVar or HGMD, and that we have not, or if it has, we haven't quite pulled it into our CAP-CLIA report because we're not yet confident enough about it. But the automated report that everyone gets when they come through, and every year that you are reevaluated and reannotated, right now is about 2,000 genes, and of course multiple variants. But really, for any of our clients in whom there's a, again, dramatic phenotype, we do have our bioinformatician really do a deep dive, and that's really been a highlight of our program. Another question, Robert, was on polygenic risk scores. And I'm actually summarizing a couple questions here. Do you think that polygenic risk scores will in some ways basically represent kind of the so-called missing heritability, or account for the missing heritability? We know that, for example, out of neuropsychiatric diseases, bipolar, OCD, have a very high, heritability run in families, but very few rare monogenic variants. And, wondering what your thought is, in terms of polygenic risk, multiple genes, hundreds, thousands of genes, each with a small effect size, causing someone to have a slightly increased or decreased risk. What's your sense of that? Oh, you're on mute, Robert.
You did a great description, overview of polygenic risk scores for anybody who doesn't know it. And just to build on that, yeah, imagine instead of one gene that causes a disease, that there's 10,000 different markers that if they're tilted in one direction, add risk. If they're tilted in another direction, protection. And imagine that just by luck, most people are in the middle with, half of them toward the risk side and half of them toward the protected side. But on the tail ends, there's a lucky few who are protected and an unfortunate few who have all those toggles switched in the risk direction. And they can be in the highest percentile of known genetic risk. But the thing to keep in mind is that for any given disease, everything we know about polygenic risk only represents at most 10 to 20% of the variance. So other things are lifestyle, diet, unknown genetic risks, lots of other things are influencing. So I think polygenic risks are very interesting, and I'm using them in my preventive genomics clinic as well. So I'm glad you're working with them. But occasionally there's a paper that I just heard about a paper today. In fact, at a seminar I was at, there was a paper that showed that when they tried to calculate polygenic risks from about 10 different companies or different, that they were all different. So we haven't quite. Yeah, it's Abramowitz et al. 2024. It's in medRxiv. And 50 PRS scores that were attempted and were quite variable. So we haven't nailed it yet in terms of how to use them, and especially how to use them with different ancestry groups. But I think it's a really important area.
That's great. Thank you. Next question is on, pharmacogenomics, Robert. What is your experience or take on, implementing that into, a typical primary care practice? Many of our clients, as you know, have their own, in some cases, concierge primary care physicians or primary care physicians, and they get our pharmacogenomics report and they're like, what the hell do I do with this? And what's your take on kind of the adoption really by our primary
Yeah.
care colleagues?
That's a hard one because we all intuitively appreciate that pharmacogenomics is valuable. And in some very carefully controlled Dutch studies in the Netherlands, there's been real demonstrations of money saving and medical benefit to using pharmacogenomics. But the way you obviously have to eventually use it is everybody's got to have some pharmacogenomic profile done. It's got to be sitting there in the system. And when a doctor goes to start you on a drug, it has to pop up and say to you, hey, wait a second, this person might tolerate a different drug better, or they might tolerate a lower dose better. You want to pay attention to this. And there's a few systems that have put this kind of thing into place. Sanford in South Dakota, St. Jude's, a few other systems. But otherwise, you had to do it manually. That means, and I'm not sure how you're doing at HLI, but in most of the concierge practices I'm working with, the doctor luckily has a small panel of
Yep.
patients, uh, and when they start a new drug, they pull open the chart and actually look at it, and they say, "Oh yeah, we should start a lower dose of statin in this person."
Right.
So that's okay, but it's not going to scale readily.
Yep. That has been our experience. We typically, if there is adoption, it's that very sort of manual process. And I think to your point, the EMR, the EHR of the future, as you're prescribing, will send up a, little red flag. Hey, doc, you might suggest raising or lowering this drug based on this person's pharmacogenomics. So
Yeah.
great. Next question. You had mentioned this a little bit, Robert, and that was very intriguing about, kind of, first of all, the percentage of folks with rare monogenic findings. And that, as you pointed out, recapitulates what we saw in our PNAS paper, which is about 1 in 6,
Sure. Yeah.
17%, right, had rare monogenic variants. And I think the question that always comes up, how actionable, right, are they? Not just in terms of, are there things you can do to sort of, better screen, but what about kind of on the intervention side? And so the question is, to what extent are, especially maybe some of the more dramatic rare monogenic variants, to what extent are they sort of truly actionable by kind of interventions, not just kind of increased sort of screening, earlier screening, more rapid screening, but in terms of interventions? What's your take on that?
Yeah. Well, actionability, and I got to write an essay on this because I've been studying this for a decade. Actionability, I don't know, like art, like pornography, like taxes. Actionability is in the eye of the beholder here. What is actionable to one person is not to another. So I've been kind of informally, nobody's agreed on this, but I've been trying to informally considering actionability to be the bigger concept where you even can prepare your life, your retirement, your insurance, what you're interested in following in the research world, but certainly incorporate surveillance, alertness in your clinician. Doctors miss a lot of stuff. So if I told you that a child had a mutation for a heart conduction abnormality, and that child had a fainting spell on the playground, you could save years of 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 red herrings by knowing that that mutation was actually there, right?
Yep.
Anyway, but the other is, as you said, is treatability. That's the other word I use, like treatability. Is there a specific treatment, a specific intervention that's known to lower risk or known to delay onset or progression? I mean, uh, even that's hard to define.
Yep.
But I will say that we picked the first 56 genes of the ACMG list with that narrow definition in mind. There was a specific intervention, and that list has now grown to 81 genes, and that 81 genes is present in approximately 4% of the general public. So, if you want to extrapolate from that experience, we think 4% of the general public is unambiguously treatable right now.
Yep. Well, that makes sense. Maybe I'll just follow on to that because there were a couple questions in that regard. I think this is maybe an editorial posed as a question, but I'll go ahead anyway. We see a lot of folks, right, with rare monogenic variants, some cases elevated polygenic risk score for some, as you might imagine, some pretty serious diseases, including neurodegenerative diseases, and we, occasionally actually have had some folks come in, with Huntington's disease or others, very serious neurodegenerative diseases. And, the one thing that we know is that even if somebody has, the same trinucleotide repeat. And we of course know there's some variability there, that there is a relatively wide range of presentation of the phenotype of the disease, of the movement disorder. Some folks presenting in their 30s, 40s, others, 60s or 70s. And it doesn't quite make sense to me that that's all because of the length of the trinucleotide repeat. And the conclusion that we're more, I think, coming to believe in, and Dean Ornish just published a paper out of UCSF about wellness interventions for folks with early mild cognitive impairment for Alzheimer's disease, not only showing a mitigation of progression, but also potentially even a reversal of some of the, cognitive symptoms. But the conclusion I think that we're coming to is that what can potentially explain this wide range in phenotype presentation, the age at which it presents, is biologic age. And so someone that, and this is maybe where there's you let off by saying this isn't much of a longevity talk, but maybe this is where there's some interface where someone who has the trinucleotide risk for Huntington's disease and doesn't present until their 70s probably has a much lower overall biologic age, whereas somebody who has the exact same trinucleotide repeat, the same genetic risk, presents in their 30s or 40s, their biologic age, their inflammation, their insulin resistance, their oxidative stress, their mitochondrial dysfunction is much older. Right. And so,
Yeah, yeah.
in our view, we think that, and just is based really on personal observation and just, meeting and treating many, many clients over the years, that nearly all, rare monogenic disease, polygenic risk is not only, actionable, but I think actionable and treatable. Because if you think about, age is probably the only disease that matters and probably underpins, all these chronic age-related diseases. So if you can modify someone's biologic age, you've got something there to intervene.
Yeah, and I wouldn't disagree with you, David. I think in my world, there's a very, very strong bias that there has to be unimpeachable evidence before you consider something treatable. So I tend to sort of reflexively take the conservative
Yep.
posture when I'm speaking. But it is my personal belief that almost everything is actionable. And I do agree that there's a lot of interface between monogenic and polygenic risk scores. They're probably additive. We haven't even really talked about microbiome and exposome and the intervening effects of exercise, diet, and so forth, which I know you do a lot with there. But I will reiterate that genomics doesn't have to be something exotic and unapproachable. I know I'm preaching to the choir here, but just so that all the people who are listening go out and talk to their people who are not part of human longevity. We can find people with low-hanging fruit, if you will, that's eminently treatable, that's life-saving. We can do that every day, and we can do it at a reasonable cost. And we're not doing it. And I think that that's something we should be doing for the future. So I know we're almost out of time, but I do want to thank you guys. I want to admire what you're doing. I want to thank you for having me to speak. And I want to reiterate that if anyone listening is interested in hearing more about our research from a philanthropic point of view, supporting it, or our company from an investment point of view, please reach out to me right away, and I'd be happy to have that conversation.
Great. Well, thank you so much, Robert, and thank you all for attending. And Robert, we're grateful for your presentation and your support for us over the years. Thank you very much, everybody. Good night.
Take care.